Welcome back to our course on principles and biomedical applications of fluorescence spectroscopy. Today we're going to be talking about fluorescence resonance energy transfer, always described of as FRET. This will be the first of two lectures on FRET. We'll cover most of the basic material today, and in the next lecture we'll do some more advanced applications of FRET. So what is FRET? FRET is a way of transferring energy from a donor molecule to an acceptor molecule. The donor has to be fluorescent. Typically, the acceptors are also fluorescent, but they need not be. Most important thing is that the acceptor molecules have some overlap. Their absorption spectrum has some overlap with the emission spectrum of the donor. So the donor emission spectrum has to overlap with the acceptor absorption spectrum. And I'll say this several times, this is not a radiative process, meaning there's no intermediate photon. It's a through space near field interaction. And that sounds a little bit exotic, but I think, we'll get to that in a moment. What is one of the characteristics of FRET? It has a distance dependence that is very well described and has been proven in many experiments. And the efficiency of energy transfer, that is the fraction of the molecule that goes from the donor to the acceptor, decreases with one power of 1/6 of the distance. That's a very steep distance dependence. So at short distances, the energy transfer is complete. If you're at half, there's a central value called the Foster distance. That's the distance at which energy transfer is 50% complete. If you get a little bit shorter than that, just going to half of the Foster distance, you're already up to 98% efficiency. Or if you go out to twice the Foster distance, you're all the way down to 0.015 in terms of efficiency, 1.5%, as compared to 50% efficiency at R0. So it's a very steep interaction. It can give you information about distances in this distance range, but if you're outside of this distance range, it's very difficult to use the FRET data. And this is what I meant, that it's much more commonplace than you might think. You have near field, you're working with near field interactions almost every day. If you have a pass card that gets you into your building, the reason it works is that it's a near field interaction. The pass card has to be near whatever the sensor is, and when it gets close enough to it, the near field interactions can happen. Of course, it's much longer wavelengths for these, and you move just a little bit away, it doesn't work. Same thing is true for inductive charging of cell phones. It's a coupling of the electric fields surrounding a molecule or a structure. So let's look at some typical applications of FRET. One that has always been stressed until about 10-15 years ago was the use of FRET to measure distances, because it has this distance dependence. So what happens with FRET? We have our Jablonski diagram back here, and molecules can go down to the ground state by a variety of processes, but FRET is really a sort of a unique, different process because you're going over to another fluorescent species. And for example, we've used this to look at energy transfer between a shorter wavelength tryptophan to longer wavelength danzyl in a molecule like melatonin that wraps up into a nice alpha helix. And we'll bring this molecule up several times during the lecture. There's two types of energy transfer. The one that we're talking about is heterotransfer. That is energy transfer between two different molecules. But there's another type of transfer called homotransfer, where the molecule transfers to itself. So if you have something like fluorescence, there's only a small Stokes shift between absorption and emission, and because of that, an excited fluorescence can undergo fret to a nearby unexcited fluorescence, and that actually results in depolarization, which we'll talk about later. Some molecules, like the Bodipes, have extremely small Stokes shifts, and they actually self-quench very easily because of this energy transfer. If the molecule has a large Stokes shift, you'll never see homotransfer in a molecule like this danzyl sulfonyl chloride, because there's no significant overlap between the absorption spectrum and the emission spectra. Homotransfer is one of the earliest observations in fluorescence, and it was examined by Jablonski many years ago, but not using biomolecules, just using concentrated solutions. And I noticed recently that homofret was being used in increasing the resolution of super resolution imaging. I can't explain that yet. I've looked at some of the papers and I still have to work on figuring that one out. So the person who sort of discovered fret is Theodore Foster, and he came up with this equation here. It's one of the equations he came up with, which says that the energy transfer is equal to the Foster distance to the 6th power divided by the Foster distance to the 6th power plus the actual distance to the 6th power. And anything raised to the 6th power becomes a big number. So you can see if the distance is twice the R0, well, I don't know what 2 to the 6th is. It's a pretty big number. So the transfer efficiency becomes very low. This is the fundamental equation for the transfer rate, and this is actually going to be very significant. So the transfer rate is dependent on distance, and it's given by this ratio of R0 to R to the 6th power, and it's preceded by the reciprocal of the lifetime. So what does this tell us? Fret depends on the donor acceptor distance. The rate depends on the distance. We already said 50% energy transfer occurs at the Foster distance, which is something that gets experimentally determined. It's a through-spaced interaction. And one of the things that I like most about fret, it is completely predictable from theory. To my knowledge, there is no example except the one that I'm going to show you, which is very specialized. If a donor and acceptor have an appropriate spectral overlap, fret will occur between those two molecules if they get close enough. So it's sort of an absolute that you can take to the bank. And this you can see fret is sort of more quantitative than our previous lectures on solvent effects, where the descriptions were approximately correct, but somewhat ambiguous because they didn't take into account the structures of the molecules. So let's look at another example here, a distance calculation from the emission spectra. We use two different molecules here. We have a molecule here with an indole group that emits around 350 nanometers, and then a long alkyl chain, and a dancyl group on the other end, which emits at around 500 nanometers. So in a typical energy transfer experiment, you want to look at the donor intensities, sometimes the acceptor intensities, but especially the donor intensities in the presence and absence of the acceptor. So here, in the absence of the acceptor, the donor is normalized to 1, and it drops down to about 1/10 of that once you put the acceptor on. And so says your energy transfer efficiency is 90%. Something you'll notice here, I'll bring this up again, is that the acceptor emission increases when there is energy transfer, decreases the donor emission. But that's a little bit harder to use. It can be used to measure the distance between the two species, but it's not as simple. So anyway, you take this 90%, and we had a known value for the Foster distance, and you can plug it into this equation and calculate that the distance is 17.6 angstroms between these two molecules. The way you solve this is you set the efficiency to 0.9, and then you can solve this equation for R in terms of R0. So this is the expected result, that they're closer because the transfer efficiency is so high. The distance between the donor and acceptor had to turn out to be lower than the Foster distance. The spectra for donors and acceptors are more complex than they seem from the first slide. From the first slide, I only put down the donor emission and the acceptor absorption. Whenever you're doing an energy transfer experiment, you're actually dealing with two molecules, and they each have a donor and emission spectrum, and it's easy to sort of get confused here. So here's our donor absorption. That's not important for the calculation of R0. And there's the donor emission. Now that's going to be important because, well, I'll come out here first. Here's the acceptor emission. But the important thing here is the acceptor absorption. And that's overlapping with the donor emission, and that gives us what's called an overlap integral. So the important thing to note from this slide, and this is almost always the case, is there's always going to be some acceptor absorption where you excite the donor. So this will show up in many of the data sets that I'll show you. So you can't cleanly excite just the donor in most experiments. Okay, so we're coming back to melatonin again. We're going to look at calculation of the donor to the acceptor distance. Melatonin can be either in a random coil or an alpha helical configuration. Melatonin, by the way, is from bee venom, and when it wraps up, when it sticks to a membrane, it wraps up into a helix that's nonpolar on one side. That sticks into the membrane, and that disrupts the cell membranes, and that's why bees can give you such a nasty sting. So in the case of melatonin, we have less energy transfer than we had for that first pair where they were only linked by an alkyl chain. We have a donor emission again set at 1. The donor is approximately 50% quenched when there's the acceptor is there, and then we have the acceptor emission out here. Now notice that these you always see some emission from the acceptor, just as I showed in the previous slide, the acceptor is always going to absorb at the where you're exciting the donor. So this acceptor actually comes from two places. It comes from direct excitation of the acceptor and from energy transfer from the donor. And we use the standard equations. They can be used in different forms. The most common form is just use the intensities, where you have the intensity of the DA pair over the intensity of the donor alone, and use that to calculate the transfer efficiency. You could also use the lifetimes of the donor alone and the donor acceptor pair. Whichever you use, you should be getting the same answer, close to 50% energy transfer. Actually, it's about 45%, and you could calculate the distance is just slightly longer than the Foster distance. So I just wanted to put in one spot all of the basic equations that we use in energy transfer. We're linking it to the Jablonski diagram, where the molecule is shifting to the other side. It's another decay path for the molecules, and then the donor provides the most reliable value. So we had our equation for the transfer rate. You can also put it in terms of the rates, the way we calculated this for quantum yields. You might remember for quantum yields, we had the rate of emission divided by the rate of emission plus the rate of non-radiative decay. But here it's essentially the same thing. The efficiency is the rate of emission divided by the rate of emission, and this is the inverse of that. That's the rate, the intrinsic rate of the donor decay. So it's the same expressions we use for quantum yield. And we just described this one about using the intensities of the donor acceptor pair versus the donor, or the lifetimes of the donor acceptor pair versus the donor. Spectral overlap. How do you determine R0? Well, here we have put that plot together for the actual donor acceptor pair we're looking at. So this is our donor absorption spectrum, which is centered around 280 or so. And its emission is at around 350. And our Danzel absorption is way out at 500. But what's important for the overlap integral is the Danzel absorption, which is this line here. So you can see we have very good overlap between the donor emission and the acceptor absorption. So now we have to calculate the overlap integral. So let's look at what is the overlap integral. Don't get bogged down with trying to think about how you're going to calculate it. Just think of it as the following. First of all, it's normalized by the total intensity. So that's the integral over all wavelengths of the donor alone. But in the numerator here, we have the donor emission spectrum and the acceptor absorption spectrum, this part here. And that's the part that we were just pointing to here. And I couldn't explain why the lambda to the fourth shows up there. So this seemingly is a big problem for energy transfer measurements, but as I'll show you in a few moments, really not. So the reason you need the overlap integral is because you need it to calculate R0. This horrendous looking equation calculates the Foster distance to the 6th power, because everything's to the 6th power. That's because there's a donor with a 10 to the 3rd power and an acceptor with a 10 to the 3rd power in terms of distance dependence. So here we have our overlap integral, and I neglected to put in the normalization there, plus this whole bunch of constants. I've never gone through this derivation myself, but why are these all needed? And I think they're simple. If this was written in a physics book, it would probably be a much shorter equation, but Foster went to the trouble of putting it into the language of chemist. And putting it into the language of chemist, he had to change his thinking of, he had to think of what units are the extinction coefficient, and they use the usual molder -1, centimeter -1. You have to know the quantum yield of the donor. You have to need to know the lifetime of the donor. There's this thing in here is just a bunch of constants, and we have to know kappa squared. That's always considered to be a problem in measuring Foster distances, but I hope I can convince you that it's not a big problem. It has to do with overlap of the donor dipole, not overlap, but like angular overlap between the orientation of the donor emission and the acceptor absorption, and that affects the amount of energy transfer. Calculation of R0. Okay. All the R0s are going to be in the range of 10 angstroms to 100 angstroms, or 1 to 10 nanometers. You're really not going to have to calculate it because there's many tables available and probably even programs on the web. R0 is insensitive to many parameters. And as I mentioned before, with no exceptions, if there's overlap, it will occur. And if you don't know R0, but you know there's some overlap, don't worry about it. Just do your experiment and see if you get any energy transfer, because you can always go back and find the R0 value later. So the bottom line here is, and I think you'll like this, you will never need to calculate a Foster distance or an overlap integral. That information will be available in the literature, and even if you get it wrong, it doesn't hurt your results very much. So, for example, the dependent of R0 on spectral overlap is rather modest. So this experiment here, it's a series of Danzel probes attached to phospholipids with increasingly longer wavelength. And the acceptor always stays the same. It's an eosin phospholipid, and that stays in the same place. So we're choosing donors that sweep across the absorption spectrum of the eosin. So let's look at the transfer integral for the top case where there's only modest overlap, and it's something on the order of 3 times 10 to the minus 14 in some units or other. And if we go to the bottom case, we see it's more than tenfold lower, so down to 10 to the minus 13. So what has happened here? Looking from the top one to the third one, the change in the overlap integral has been 7-fold when you calculate them. But because you're always taking the 6th root of the overlap integral, this is R0 to the 6th. So if you want to calculate an R0, taking this to the 1/6 power, it's only 20-25% difference. So the R0 value is not very sensitive to the absolute value of the spectral overlap. And for those of you who like to do math here, if you take this value and take the 1/6 root of it, you'll see that it's pretty close to this ratio down here. Here's the thing that people worry about. and it's called the effect of the orientation factor. So you have to imagine two molecules, a donor acceptor in space, that have transition moments in particular directions for the donor emission, the acceptor absorption, and the transfer rate is going to depend on something called kappa squared, which is an outrageously complex bit of geometry, trigonometry, I guess. But the final analysis is kappa squared can vary from 4 if the dipoles are aligned, end to end. If they're parallel, it's 2, and if they're perpendicular to each other, it's 0. So that gives you the bad news that there can be a lot of fret or no fret at all, depending on the orientation of the donor and the acceptor molecules. That bothered people for a long time, but it's not really a problem. Here's the one case. This is the only case I found in the literature where you can see an effect of kappa squared on energy transfer. So they used this porphyrin molecule here, and they attached an anthracene to the end, and the dipole moment is across the short axis. So this is one of the cases where kappa squared is 4. So if kappa squared is 4, transfer from the anthracene into the porphyrin unit should be very rapid, and that's exactly what happens. The donor control is the green emission up here. With kappa squared is 4, you essentially see no anthracene emission. So these clever researchers, I'm sorry for the double set of arrows here, but these clever researchers then put the anthracene along the central axis of the acceptor molecule. So now we have the dipoles are perpendicular to each other, and that predicts kappa squared is 0, and by golly, that's just what happens. You see now, with the kappa squared is 0, the anthracene emission remains high. There's much less or no energy transfer into the acceptor because the kappa squared is 4. But these molecules are completely symmetric, and plus they have another advantage. No matter which way the anthracenes rotate, they're always going. The transition moments are always going to remain the same. So that's why this experiment worked. We have not yet seen a case where this has caused a significant error in any measurements of distances in proteins. And this is because if you look at the anisotropy decay of the tryptophanes, which are typically the donors, in any protein, they always have a fast component and a short component. The fast component is due to the segmental motions of the tryptophan residue, and then there's a slower motion which is due to overall rotational diffusion of the protein. But this very rapidly randomly just eliminates the effect of kappa squared. And with most of the labeled molecules that you'll deal with, it's even randomized faster than this, because if you're going to label a protein, you're going to label it on the surface. It's going to be a water-soluble fluorophore, both for the donors and the acceptors, so they're always going to have a significant amount of mobility. So kappa squared does not enter the case. As I remember, a researcher at Hopkins compared the measured R0 values and the ones from the crystal structures for around 20 or 30 proteins, and there was no significant discrepancy about that. So you can trust these distances. There are many experiments where you really don't need to know the R0. And basically all you need to know is how much energy transfer is occurring or not occurring. So here, this is actually quite an old experiment. We have a fluorescene labeled oligo, single-stranded, and a rhodamine labeled oligo, single-stranded. The donor and the acceptor are positioned so they'll be close together when it forms a double helix. And what you find is pretty much just what you might expect. If when the molecules associate, the donor emission quickly drops because it's going over into the rhodamine and being removed by fret. Or if you heat it up and dissociate it, the donor emission increases because it's moving away from the acceptor. Now, there's an important thing to notice from this experiment. This would probably be done in a cuvette, couple of milliliters of solution. When they're in the cuvette and they're just dissolved in the cuvette, they don't transfer energy anymore. They have to be very close to transfer energy, so it's only when they're in this double helical form. But the moment they dissociate, they stop transferring. And what this says is radiative transfer, meaning forming a photon that gets emitted and that gets reabsorbed by the acceptor. That just doesn't happen under any reasonable conditions. It's the hybridization that has resulted in the high local concentration of the acceptor relative to the donor. But this radiative transfer, I can't think of an experiment where it's been seen. So that was for DNA. You can also use the energy transfer to measure protein associations. And this is nice because it's sort of a wavelength ratiometric thing. So you have a donor and acceptor labeled protein, and if they dimerize, the donor goes down, the acceptor goes up. If they dissociate, it's the other way around. Donor goes up, the acceptor goes down. So let's look at an actual example of this. So this is for a protein named thymidylate synthase. It's put methyl groups on uridine residues to create thiamine residues, and it's involved in DNA methylation. So the authors here, it exists as a dimer. The authors here labeled the subunits with different fluorophores to measure the association. And it turned out that the results were a little bit complicated, but they're also instructive. So here's donor and the acceptor listed, and here are their absorption spectra. Ignore the gray one, that's for a mixture, but you can see the fluorescene absorption and the rhodamine absorption, and so there you expect transfer to occur. But the problem with the experiment is that he wound up with a mixture of dimers. Some of the dimers were fluorescene labeled dimers, and in that case there's no shift in the there's no energy transfer. But if you get a fluorophore with both probes on it, listed as 2P, well, then you get significant energy transfer. So you can, well, I would suspect it's always going to be an equal distribution because the subunits are identical. You can also see this in the absorption spectra. In the absorption spectrum of the fluorescene labeled one alone, you'll see the fluorescene absorption spectrum, when you have both probes there, you see the absorption of the rhodamine plus the absorption of the fluorescein. Remember, this was an excitation spectrum. Let's see, and what says an absorption spectrum, you would see this also in an excitation spectrum, where you're scanning the emission intensity while you're changing the excitation wavelength. So you can see the fret in the absorption spectra. The time-resolved decays, unfortunately the authors didn't put the impulse response function here, but the time-resolved decays indicated that the fret must be rapid, otherwise there should have been a rise time in the decays. But how can you see the subunits dissociating? Well, you just dilute them, and as you dilute them with the highest concentration, you see a lot of the rhodamine due to energy transfer. At the lower concentrations, they dissociate, and you don't see energy transfer anymore. You could see this way, and in a graphical way, where you see the energy transfer efficiency increases with protein concentration, and the anisotropy is going up. And I have to assume that it's going up because of the increase in the molecular weight of the dimers relative to the monomers. Oh, notice in that previous experiment, we didn't have to know anything about R0. All we had to know was that energy transfer could occur. Normally, you're not going to do a X-ray experiment on a lipoprotein. So if you want to learn something about the structure, the fluorescence can be helpful. So for high-density lipoprotein, otherwise known as the good lipoprotein, there were two possible structures. It was known to contain two peptides, identical peptides, but it wasn't known whether they were wrapped as a bell, wrapped around the lipids in a circle, or if it had a picket fence conformation. So what was done here is the authors got the peptide, labeled it at precisely the same site, one with a donor, one with an acceptor, and put both of those into formation of the HDL complex. And they reasoned that if this belt structure were correct, they should see energy transfer. If the picket fence structure was correct, they would not see energy transfer. And that's basically what happened. They did see energy transfer and concluded that the belt structure was correct. FRET has also been used with molecular beacons. Molecular beacons are those DNA fragments that have a loop that allows a complementary region of the DNA to come together. And if in the sample it finds sequences that match here, well, that breaks apart the molecular beacon and the energy transfer is turned off. And that's exactly what you could see here, is that as they added the complementary oligo, the donor intensity went up and the acceptor intensity went down because they were removing energy transfer. This type of experiment is very frequently done with what are called black hole acceptors. Dabsil is one. I should have put the structure here for you, but well, it's on the figure. It's non-fluorescent. A acceptor molecule does not have to be fluorescent; it just has to absorb. And this type of a system is frequently used in PCR assays, because as you form more and more of the fragment, you break up, that's target fragment, you break up the molecular beacon and you see higher intensities. So this is used not in a structural sense, but just to measure the extent of PCR. And I think this is the last example I have for you. Oh no, there's two other ones. This is if you haven't heard of them, these are chaperone proteins that exist in bacteria. They used to be called heat shock proteins. What this system does, it helps proteins fold back into their native form. So in order for it to be active, it has to be completely together. The top of it has to be on the lower part, and when that's together, them coming together is catalyzed by ATP, and the proteins refold inside this hollow cage is the bottom structure of it. So if you put these two proteins, we labeled one with fluorescine, another one with another aminoanthracene type fluorophore. If you put the two separate subunits in solution, you get two separate emission spectra, one from the top subunit, which is the longer one, I believe, and one from the bottom subunit. And if you mix them both in there, it's just the sum of those two spectra. So these are not interacting at all. But if you just put a little bit of ATP in there, they bind together almost immediately, and the donor emission almost disappears. So that shows they're binding together, and the kinetics of binding can be seen by the growth of the donor or the decrease of the acceptor. Well, that's actually in the dissociation side, which I presume all the ATP is used up. Incomplete labeling. So this has sounded easy so far, but incomplete labeling is a significant problem in energy transfer, because if you have a system where the donor is quenched extensively, let's say 90%, and you have 10% molecules that don't have an acceptor by them, well, they're going to give an equal amount of emission. So I remember when we were first doing some of this work with distance distributions, which will be in the next lecture, just a little bit of unlabeled material can totally mess up the measurements. We have the equation here for how to correct for it. And here's the complete, when there's no, when they're completely labeled, and if it's incompletely labeled, well, then there's more donor emission because of the acceptor-free molecules. I've stressed all the way up to here the value of using the donor emission. And that's because it's just more reliable. You can calculate the amount of energy transfer from the change in the acceptor intensity. The problem with that is you have to correct for the amount of acceptor intensity that you would have got even if the donor wasn't there. So this correction factor is expressed in this equation, which has the extinction coefficients of the donor and the acceptor at the excitation wavelength, the fracture amount of labeling. It's pretty, it'd be difficult to derive out of this setting, but this is always a problem if you have incomplete labeling. Lanthanides. Lanthanides are also frequently used as FRET donors. They have 2 electrons in the outer shell, which we described already, and that shields them from the outside world. Their behavior in energy transfer is quite interesting. They have very low extinction coefficients, so they can't be excited at all. And the way they are excited is by chelating with a fluorophore. And because it's right next to the europium or terbium, you can excite that. And from there, you can get energy transfer to an acceptor. So this was done here with an oligo. So the decay for the donor alone, we see is up in the millisecond time scale. If we look at the sensitized emission, that's on a much shorter time scale. And of course, there's a mixture of molecules, as there always is. So some of the molecules have the acceptor next to them, and some of them don't have the acceptor next to them. But it's very interesting that if you look at the emission spectra, you might remember that the lanthanides are structured, and you look at the cyanine 5, I guess that is, emission, you find out that it has this enormous lifetime of 240 microseconds, which is ridiculous because we know all of these fluorophores have nanosecond decay times. But this has happened because the acceptor is being excited continuously from the donor, so it appears to have this long lifetime. This is something that always bothered me. Why is FRET even possible with lanthanide donors? You would think in all the cases we were looking at with nanosecond fluorophores, okay, energy transfer occurs in nanoseconds. So if you have something that has a millisecond lifetime, well, certainly that energy transfer is going to be complete. So I was always intrigued by the fact that you could measure energy transfer from the lanthanide lifetimes. And I finally looked down and figured out why that is. And the reason for that is that these two terms here, the quantum yield over the lifetime, is essentially the radiative rate of the lanthanide, or whatever the emitter is. So the transfer rate is always proportional to the intrinsic radiative rate of the fluorophore. So the transfer rate with the lanthanides is way down in the inverse of the milliseconds, because it's related by this ratio. So that's the end of part 7a. I think I might have caused a little confusion at the end, but maybe if you listen to it more than once, my mixed up words will clear up a little bit. I thank you all for visiting and hope to see you at our next lecture. Bye.